Hello, my name is Fiona O'Donovan, and today I will present my open innovation in science idea. Growing evidence indicates that maternal stress impacts the physiology and the psychology of the fetus. Elevated maternal stress during pregnancy has been associated with structural and functional changes in the offspring brain, and these changes have been associated with a range of behavioral problems that can manifest in infancy, develop throughout childhood, and follow an individual into their adult life. In recent years, advancements in brain imaging methods are helping us to slowly start to uncover the mechanisms through which maternal stress can act to bring about these changes. However, there are roadblocks slowing down our research, preventing us from going forward as efficiently and quickly as we would like. The timeline from when we acquire magnetic resonance imaging to when we finally get a result from the acquired data is long and can take years. A considerable amount of this time is consumed by the process of processing the data. One such process that takes a large percentage of the time is quality control. But this is crucial in ensuring the highest possible quality of our data. During this process, a researcher must go through many different types of imaging data and determine which data can be used, which data needs to be fixed using algorithms, and which data cannot be used. As all these processing pipelines have been developed for adults, this process gets a lot more tedious when applying it to infants and children. After spending hours of time doing this work myself, I began to think of a solution to this using the open innovation science techniques. By creating an online platform, such as on Zooniverse, everyone will be able to take part in a cutting edge project that will be able to help me at the same time. A tract, like the ones that were shown in the previous image, will be presented to the participants, and they will be able to decide if the reconstructed tract looks bad, okay, or good. As this is as this does require a certain level of knowledge, I will first provide the participants with a brief but informative training, as well as some test examples before they themselves can begin the process of quality control. Once trained, the participants will just have to rate the tract before continuing on to the next tract until they are finished the sample set. By doing this, I hope to increase productivity and efficiency in my research. For this work to work best, and for the people who are involved to benefit the most from it, I would like to co-create this process with a team of clinicians, engineers, computer scientists, and MRI analysts, as well as the users themselves. Additionally, so the users benefit throughout the process, I would like to use this platform as a method for disseminating information about the topic of stress and potentially at a later stage, the results from my studies. In this way, not only is the productivity increased, but also the user will gain more than just a good feeling from partaking in the project. I want to get everyone to get involved in this project, but in particular, I will aim this project at parents via online forums, as well as students themselves who might be interested in MRI research in the future. If this proposed method is successful, I would like to additionally use it as a method to identify the best pre-processing pipelines. In recent years, many different groups have developed processing tools. In general, groups will have a bias towards their own tools. As quality control is so time consuming, we as researchers do not have time to compare these tools. In addition, I also hope to use this method as a way to identify the best pre-processing pipelines. Through this open quality control tool and by recruiting a large number of users, I hope to use that the power of numbers will be able to identify which are the best pipelines to remove any potential bias that might be generated by the groups creating the pipelines themselves. Here are the major milestones of the project. I will begin with the co-creation phase, followed by a short tester phase to ensure the tool functions correctly. While the program is available and people are partaking, I will do some analysis to ensure that the task has been completed to a sufficient standard. Once complete, I will then proceed to analyze the data and based on the participants' feedback, further QC the bad data. If this project is successful and participants want to continue with the project, once one track is complete, an additional training can be made for another track and participants can start quality controlling a new track. If participants continue to use the platform, this method could become an option for other MRI researchers who are working with infants and children and MRI data. Getting to the answers to the research questions that we want to answer requires large studies with hundreds of thousands of subjects. However, it is of the utmost importance to continue using the highest quality of data we can to ensure that we are doing precise science. By implementing a method like I have described in this presentation, I believe we'll have the opportunity to perform correct quality control efficiency in these large study populations. I hope that this will lead to the answers of my research questions faster and help me to improve healthcare and well-being of infants and children. Thank you for listening.